Dear student, welcome to the Medical Genetic course. Today's topic is cytogenetic and molecular techniques used to investigate genetic diseases. By the end of this video, you are expected to know the clinical indication for genetic testing, explain the difference, method, and techniques used for different genetic testing, and describe cytogenetic techniques used to investigate for chromosomal abnormalities. You will also know the different types of probes used for FISH analysis. You will know the importance of PCR and real-time PCR and their uses in molecular genetic diagnosis. You will also distinguish between different types of sequences. There are two types of genetic testing. It depends on the genetic diseases that we wanted to investigate or to confirm. The first type is chromosomal genetic test. In this lecture, I will explain karyotyping and fluorescence in situ hybridization. The second type is molecular genetic test, and in this lecture, I will cover PCR and sequencing technique. These molecular and cytogenetic techniques have been applied to identify genetic mutation leading to diseases. Accurate diagnosis of disease is essential for appropriate treatment and management of patients. Different techniques are available to perform genetic diagnostic tests on patient specimens, both for inherited and acquired genetic anomalies. Genetic mutations can be either constitutional, which means it present in every cell of the affected person. It means also it is inherited. Or genetic mutation can be restricted to specific type, which is called somatic mutation. There are indications for analysis of genetic alteration. There is a prenatal test which is done for fetuses during pregnancy. It depends if the maternal age is advanced or if one of the parents carry balanced chromosomal rearrangement or if they noticed abnormalities in fetuses during ultrasound during pregnancy. There are also tests that are done following birth. For example, if there is a malformation identified in newborn, or if there is newborn screening test for metabolic disorder after birth. There are also indications for genetic tests in children. For example, if they have an X-linked genetic retardation or developmental delay, if they exhibit any syndrome that are caused by chromosomal abnormalities, or if they have suspected monogenic disorders. On the other hand, an indication for genetic testing in older patients if they have inherited cancer within the family, or if there is monogenic diseases or neurodegenerative disorders. There are also indications for analyzing the acquired genetic alteration. For example, diagnosis and management of cancer, detection of the acquired mutation and cytogenetic alteration that occurs in tumor. Also, an indication for analysis of acquired genetic alteration is management of infectious diseases. To test genetic material for medical reasons, samples are collected from blood, urine, saliva, body tissue, bone marrow, and hair. For prenatal testing, genetic material can be also collected from amniocentesis or chronic villus sampling. Genetic materials can be submitted in a tube, on a swab, in container, or frozen. Once received in the lab, the genetic material is separated and removed from the sample to proceed with the genetic testing. As I mentioned before, different techniques are available to perform genetic diagnostic tests on patient specimens, both for inherited and acquired genetic anomalies. First, we will talk about cytogenetic techniques, which focus on chromosomal abnormalities. The most common cytogenetic testing is chromosome banding, which help in determining the patient karyotype. Chromosome banding refers to alternating light and dark region along the length of a chromosome, produced after staining with a dye. It includes G banding, Q banding, and R banding. The most common used dye is Giemsa banding, or G banding. This technique helps determining the abnormal number of chromosome and structural abnormalities such as insertion, deletion, duplication, translocation, or inversion. However, the resolution of this technique is very low, because it doesn't identify microdeletion in the chromosomes. Another important cytogenetic technique is called fluorescence in situ hybridization, or FISH. It uses fluorescence DNA probes to target specific chromosomal location within the nucleus, resulting in colored signals that can be detected using fluorescence microscope. It allows reliable detection of small deletion. FISH analysis can produce a superior resolution compared to G-banding chromosome analysis. FISH can be used to detect chromosomal abnormalities which are not identified by routine karyotyping. FISH can be used to test for missing or additional chromosomal material as well as chromosome rearrangement. The FISH technique can be extended with multiple colors to detect several possible alterations of chromosome number. There are several probes used in FISH, which depends on the genetic test that needs to be done. For example, whole chromosome paints can be used to stain the whole chromosome. However, the centromeric probe is more specific to the centromere region. And locus-specific probe is for specific locus or region in the chromosome, whereas telomeric probe is specific for telomeric region in the chromosome. So I gave you two examples for the most common techniques in cytogenetics. In the next few slides, I will talk about the second type of genetic testing, which is the molecular test. Molecular genetics refers to all diagnostic methods that examine genetic changes in extracted DNA. How do we obtain the test material? While cytogenetic analysis typically requires cells that are mitotically active, molecular genetic testing only requires DNA. Other common test material are fibroblast, oral mucus from saliva, and amniotic fluid cells or fetal cell from chronic villi biopsies in prenatal diagnosis. The first and the most common method used for molecular genetics test is PCR, which focus on amplification of small DNA fragments. After PCR, the amplification products can be further examined with respect to sequence, size, and quantity. PCR involves using short DNA fragment called primers to select a segment of the genome to be amplified, and then multiple rounds of DNA synthesis to amplify the segment. First, it includes denaturation of the double strand. Second, the hybridization of the primer, which is called annealing to the DNA. Third, extension of the primer and polymerization. At the end of this step, the synthesized DNA region that we want to test is doubled. By repeating this process through many cycles in the PCR machine, an amplification of targeted DNA is achieved, as newly synthesized strands are also amplified. What is the advantage and usage of PCR? PCR provides convenient and efficient means of making millions of copies of short DNA sequence from very small quantities. The advantage of PCR include the speed and the ease of use. This technique is now widely used for assessing genetic variation, for diagnosis genetic diseases including mutation identification, detection of small deletion, and triplicate repeat expansion. Another method used is real-time PCR or quantitative, which is a method used by which the amount of PCR product can be determined in real time in a quantitative way. The advantage of real-time PCR is that it generates rapid results, with some test result being available within 1 hour of sample being taken. It is also used in genetic diagnosis and investigation for gene expression. Real-time PCR is also used to monitor the frequency of cancer cells bearing specific genetic lesion, such as the level of PCR APL mutation in patients with chronic myeloid leukemia, or the infectious load of certain viruses such as HIV or COVID-19. Another important type of molecular testing is DNA sequencing. The idea of sequencing is to determine the order of the four chemical building block spaces that makes up the DNA. The sequence reveals the kind of genetic information that is carried in particular DNA segment. There are two types of DNA sequence. Sanger sequencing, which focus on a single gene. The second one is high-throughput sequence, which covers the whole genome or just the exon part. One of the methods for detecting mutation in a single gene is called Sanger sequencing. Sequencing allows identification of mutation and prediction of their effect on the amino acid sequence and the structure of the coded protein. Sanger sequencing is considered as the gold standard for sequencing determination. Here, the amplified DNA is mixed with a DNA polymerase, a DNA primer, and 4 dead-end nucleotide A, T, G, and C. It's labeled with different fluorescence tags. The reaction produces a series of DNA molecules labeled with tags corresponding to the base at which the reaction stopped. The advantage of this technique is that it is fast in determining the change in the DNA sequence in a single gene. High-throughput sequencing methods allow the sequencing of millions of individual DNA molecules. There are two types of high-throughput sequencing. The first one is whole genome sequencing, which sequences the whole genome, the coding and non-coding DNA. The second type is exome sequencing, which is limited to the coding sequence, the exon. These techniques enabled researchers to identify mutation in the functional region in the genes. Whole exome and whole genome sequencing have been used to detect de novo mutation in a variety of conditions, especially for undiagnosed rare diseases. Finally, these are the references used for the lecture. You can also go back and see the video How Does PCR Work? In summary, with that, I end this topic, which included the following: clinical indication for genetic testing, different methods and techniques used for different genetic testing, cytogenetic techniques used to investigate for chromosomal abnormalities, different type of probes used for FISH analysis, importance of PCR and real-time PCR and their uses in molecular genetic diagnosis, lastly, different types of sequencing.